Thank you, everyone. It's an absolute pleasure to be here this evening. And Lorraine and I will be presenting an overview of the Zero program, but also a real-life illustration of the program as well. So this slide is probably a little small, but it's an overview of the Zero program and how it's improving precision medicine in children with cancer. So if I take you through the journey of a child will get sick and present at a hospital. And at the hospital, they'll be diagnosed with the cancer. So while they're at the hospital, they'll get a medical procedure and obtain a sample of their tumour. And that tumour will end up at the Children's Cancer Institute. So once it arrives at the Children's Cancer Institute, we then perform an in-depth genomic analysis. So while that's ongoing, the patient continues with standard of care. So once the data comes back, we then present the findings to a multidisciplinary tumour board. And basically what that means is you've got roughly around 80 professionals dialing in across Australia. And this is where we discuss and review the findings for each of the cases, so each of the children that are going through our program. And effectively what we do there is also share those findings with the treating doctor as well. And with the hope that it'll alter potential treatment for that child. If the patient has enough material, we then perform preclinical studies. And effectively what we do is drug screening on the actual patient samples in the laboratory, but also we have them in mouse models as well. So that also gives another indication as to how the cancer is behaving and potential drugs that could be used for the patient. So again, we repeat the process and we bring that to the multidisciplinary tumour board. Again, the results are reviewed, discussed, and then shared with the treating oncologists and with the potential of refining treatment for that child. So the next couple of slides, I'll take you through the patient journey, the patient sample journey. So basically, we get a blood sample and a tumour sample from the patient. We then isolate what's called DNA and RNA, and then we perform sequencing. So effectively, we do this in-depth genomic analysis. And what that means is we can perform whole genome sequencing, RNA sequencing, methylation analysis. And really what that means is that we're using state-of-the-art technology to have insight into the patient's tumour. So then once we have the data, this data is also reviewed through another group, which is the omics curation team. And the omics curation team consists of cancer and germline experts. And this includes bioinformatic specialists, computational biologists, curators, molecular geneticists, oncologists, clinical fellows, you name it. So in this meeting we have every Friday, and we typically have around 30 to 40 people dialing into this meeting, where we discuss on average around 8 patients in 2 hours. We come to a consensus on what's of clinical importance, and we bring that forward to the multidisciplinary tumour board. Again, that meeting is weekly, and on average we have around 80 professionals dialing in to those meetings weekly. So again, from that meeting we come to consensus of what's actually going to be on the patient report, and that report goes back to the treating doctor and ultimately for the patient. And on the right-hand side you can see just a map, and it's basically what Michelle was saying. We collaborate with all 9 paediatric oncology units here in Australia, but we're also tapping into our international medical research institutes as well. So this is over in Europe, the US and Canada. I'll pass on to Loretta.
Thank you. Thank you for that. Can you hear me? Yeah, okay. So this is the journey of one of our patients who was enrolled on Zero. So Baby Hannah is actually a made-up name because of patient confidentiality, but I love to have a name put to it because it's more personal. This way? Okay. So this baby actually was bothered by seizures all her life, even at the newborn period. So at 4 weeks of age, she had more seizures, and then an MRI was done. So as Richard had mentioned to you just now, you know, a small baby with MRI need a special machine. So an MRI was done, and it found a really large cystic cavity in her brain. So you can see on this MRI picture there, I don't think you need a radiologist in the audience to say something is not right, because one side of the brain looks like normal brain, the other one just had this big, you know, ring in there, which is a big cyst. And that white line surrounding that big hole or cyst is actually possibly lined by tumor. So this baby actually had her first brain surgery at 4 weeks of age. Imagine a tiny baby having brain surgery at 4 weeks of age. The tumor came out, and then our pathologists looked at it under the microscope. So the pink and purple dots over there are actually representing what a pathologist will actually see under a microscope. And unfortunately, the result came back is that we're now dealing with an infant malignant brain tumor, essentially from a baby who was actually born with a malignant brain tumor. So at that point, she was actually started with chemotherapy at 2 months of age, and then by 4 months of age, you know, she actually went back for a second round of brain surgery. Now, and then afterwards, she actually continued on chemotherapy for her malignant brain tumor, an aggressive brain tumor, from the age of 2 to 9 months. Imagine, you know, she essentially spent the first 9 months of her life, you know, having surgery and getting chemotherapy for a brain tumor. Despite all that, you know, she's still left with residual brain tumor, which was just slowly growing all the time, and it probably would have grown even faster without chemotherapy. So then, what's the treatment options for baby Hannah then? You know, now we've tried chemotherapy for now 7 months. Are there any other options? So thankfully, her tumor from the first surgery was actually analyzed by Zero. So we, as Paulette has said, we sequenced the tumor, and you know, we actually discovered what we call a gene fusion. So it's kind of like a mutation, but you know, 2 genes basically got separated or got smashed apart and then get put together. Anyway, what it really means is that there's actually potential drugs that's available to so-called target that particular gene fusion. And as Paulette has just described, we actually, if we have the opportunity, we would actually put the tumor cells through what we call a high-throughput drug screening process. And the picture here just shows one of our fascinating machines that allows us to do this type of drug screening procedure, where the tumor cells are essentially treated with over 150 different drugs, and they're all like put on little wells in the plastic dish, and then you just add 1 drug to each of those wells. I think you need to see it in operation to figure out how fascinating this is. Now, there are 150 drugs, and actually within those 150 drugs, there are 6 drugs that potentially can be effective or can target this particular gene fusion that we find in this baby's brain tumor. And we were very pleased to find out that the tumor cells was actually, out of the 6 drugs, most sensitive to 1 of that drug. And so what happens is that, you know, as I said, this baby has gone 7 months of chemotherapy, which is not really effective. So then the next step, actually what happened to this baby is that she was actually started on this targeted drug, you know, through enrollment in a clinical trial, which was made available to her. So unlike chemotherapy, which is given intravenously or through a drip or a central line, this is actually an oral drug just given twice a day. It even comes in an oral suspension. So little baby can't swallow tablets, right? So the drug company actually made a liquid out of that. And then she did an MRI after 4 months, you know, or 3 months. And, you know, surprisingly, or we were very pleased about it, is that she actually responded after just 3 months of that targeted therapy, and she's currently 6 months into that treatment. So then people ask, well, this is just one of the so many children. How about if you put everyone together? What does that really mean, you know? So I have to say more than 1 child has actually, you know, benefit from Zero. So as Michelle has actually described what is going to be reported in the very near future in one of the top medical journals, is that, you know, high-risk children analyzed by the Zero Precision Medicine study and then get what we describe now as precision-guided treatment over half of them will actually benefit from that treatment. And this is actually a very substantial proportion of children who would actually benefit from zero and getting precision medicine type of treatment.
So I guess what Richard said, how do we do this better? And so we're hoping that the MCCCC is the place to allow this collaborative partnership to bring together the clinicians, the clinical staff, researchers, academics into this new and integrated facility. It'll help facilitate combining research and clinical care and support training and education opportunity for researchers, clinicians, and to the wider community. It's also going to allow the seamless integration of doctor and scientists to improve the use of patient data from the lab bench to bedside more efficiently. So in the aim, it's basically to improve health outcome for children. This is just an acknowledgement slide of all the group that basically touch at every point each child that comes through our program. And also our Zero supporters. Thank you.